Okay. So this is just to illustrate the size differences, the scale. So this is a logarithmic scale. So every mark we're increasing by a power of 10. So here's a way to kind of get this concept in your mind. Here's an ostrich egg, but we're magnifying this mark by 10 to get to the size of a human. So most of what we see, our prokaryotic cells tend to be around 1 micrometer. So like this is like really, 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 really little, right? Our eukaryotic cells tend to be somewhere between 10 to 100 times that size. So our eukaryotic cells are much, much bigger than prokaryotic cells. And then we get these things like a human egg and a frog egg and a chicken egg and an ostrich egg that are actually, we can actually see with our naked eye. Human eggs, they're hard to see with our naked eye because they're really little, but you can't do it. So, okay. So linking this concept back to what we studied with our little surface area to volume ratio, why do we see our cells being really little? Like we do have, I mean, an egg is a single cell, but there's a lot of nutrients and stuff that's already stored in there. So we don't need to get a lot of nutrients in. They already got it. Why do we see our cells be kind of between 1 and 100 micrometers? Like really little.
Because of the surface area to volume thing?
It's all about the surface area to volume ratio. Comparing our big cube and our little cube, right? We saw a huge difference in that surface area to volume ratio with the smaller one had much higher surface area to volume ratio. It's all about the ratio, baby. Okay, I think that's what I want to say about that. And then we don't really often see cube cells very often. They're either circular or they're cylinder. So like here's some fungal hyphae, which is basically just a bunch of cells stacked end to end. Now my markers are gone now that I want to use them. But if you guys can imagine a bunch of whiteboard markers stuck end to end to end, right? And you get this big long cylinder. That's basically what a fungal body is. And they need a really large surface area to volume ratio because the way that they eat is they just like pump out a bunch of enzymes into the environment. It breaks stuff down and then they suck it back into their bodies. They have external digestion. We have internal digestion. But even this really high surface area to volume ratio sometimes is not enough for what they need to do. So here, this is this big kind of structure is a plant root, so we can see the xylem and the phloem, the vascular tissue of the plant. That's how it moves stuff around. And then all these little squares are the plant cells. And these kind of little black dots are even more finely, highly branched fungal hyphae, because this type of fungus lives in a symbiotic relationship with the plant roots, where the plant gives the fungus sugar that it makes through photosynthesis, and the fungus gives the plant water and nutrients that it pulls out of the soil. So for that nutrient transfer to occur, we need a super duper high surface area to volume ratio. And that's what these little arbosules, that's what they're called. So this is just some application of those principles that we just investigated. Okay, but in our human bodies, we see this too. So this is a micrograph of a biopsy of a lung cell. So you can see here all this folding is increasing the surface area in the lung cell. Why do we need a lot of transfer in our lungs? For respiration, right? We got to get oxygen in, carbon dioxide out. We need a lot of transfer. Same with our stomachs. So here's the stomach lining of a human. We can see all these finger-like projections. Why do we need a high surface area to volume ratio in our guts, in our small intestines?
For nutrient absorption.
Yeah, that's where we absorb our nutrients. So we want to be able to access those nutrients really easily. We do that by increasing surface area. Okay. Cool. All right. This whole section is 3 slides. Prokaryotic cells. Yeah, baby. So here's what we got to know about the structure of prokaryotic cells. The DNA is in the nucleoid region. Remember that prokaryotes don't have a true nucleus. But what they do have is a chromosome that kind of tends to bunch up and kind of hang out in one little spot. So it's kind of like a nucleus, but it's not really a nucleus. So we call it a nucleoid because it's nucleus-like. So anytime you see that oid, you're like, oh yeah, kind of like that thing, but not really that thing. Okay, so there's our nucleoid. It just is that one circular chromosome that's kind of been twisted and bunched up. Okay, so let's find, before I move on, let's find the things that they have in common with eukaryotic cells. We got our plasma membrane, right? Here it is. We've got these extra layers on the outside that we'll talk about in a minute. We got our cytoplasm. This is just the cell juice. We got some ribosomes. And then we got our DNA. Okay. Okay. Okay, so here we go. Like, oh, I just said all that stuff. Okay, so here's our plasma membrane again. That's our phospholipid bilayer. On the outside of that plasma membrane, we have a cell wall. This is something that animal cells don't have. Animals are the weirdos because they don't have cell walls. Basically, every other kind of cell does have a cell wall. And then on the outside of that cell wall, we have this capsule, which is for protection and to protect from dehydration. And so when we're like, all right, everybody, you got to wash your hands for 20 seconds. It needs that, the soap needs that time to penetrate the capsule and the cell wall of the bacterium.
You know that word, peptido.
Peptidoglycan, just, no, it's just protein sugar stuff. It's, yeah, I maybe know the context of it, but if you, yeah. So really what I want you to focus on is cell walls and a capsule, right? So polysaccharide, we know that that's a complex carbohydrates. So now we're starting to see like all this stuff that kind of seemed amorphous. Now it's starting to make connections. So now we're bringing it in to actual living things. Okay. Some prokaryotes have a flagellum, or the plural form is flagella. So that's how they get around. Sometimes they'll have these pili, which are used for docking on and used in conjugation, which is kind of like sex, but not actual sex. Prokaryotes don't have, they don't do sexual reproduction like eukaryotes do. They do something that's similar, but it's not the same. And then they have these other things that stick off the cell wall that allow them to attach to their host if they are parasitic. Okay, and we already talked about this. So prokaryotes, Our super teeny tiny eukaryotes are sometimes, like way bigger than those guys. 10 to 50, times bigger. They're like huge. So why again is it advantageous for cells to be small? Diffusion, right? We can just rely on passive diffusion. We don't have to pump stuff in and out of the cells. We don't have to use energy. We can just use the natural order of things. Okay, that's prokaryotes. Now we can start talking about eukaryotes. And these guys are a little bit more complicated, a lot more complicated. And so there's a lot more to it. So what we're going to want to be able to do is describe the structure of eukaryotic cells. We're going to compare animal cells and plant cells, more Venn diagrams. We already know what the plasma membrane does because we spent a whole week talking about it. So you guys should be able to check that one off. And then what are some major organelles and what do they do in the cell? All right. So the biggest differences between prokaryotic and eukaryotic cells, they have a membrane bound nucleus, right? That's where their name comes from. The prokaryotics are before nucleus, eukaryotic true nucleus. So boom, we got a nucleus. That's the defining factor. We also have in eukaryotic cells that we don't see in prokaryotic cells is a bunch of these other organelles that have a membrane. So in prokaryotes, we see ribosomes, but ribosomes don't have a membrane around them. So here's some of our membrane-bound organelles. We got our nucleus that has this thing coming off of us, an endoplasmic reticulum. We've got mitochondria that has a phospholipid bilayer. We got Golgi apparatus. We got chloroplast. We got all sorts of organelles that have membranes. And then the other difference is that in our prokaryotes, we saw that one circular chromosome that kind of bunches up. But here we have, in eukaryotes, we have several rod-shaped chromosomes, right? So we'll talk about how those are formed. Okay. Oh boy. We're going to break this down. This is not a slide. We're going to go past it real fast. Okay. All right. Organelles. This means little organs. It is like, well, so and so each of these organelles does a job, just like your lungs and your liver and your stomach and your kidneys all do a job in your body. Each organelle has a different function. And I cannot think of any interesting way to go through this list other than to say, okay, let's make an analogy of a cell city. And so then let's use something that we're familiar with, how a city functions, to talk about how cells function and what each of these organelles do. So this is going to be our little analogy. So we already talked about plasma membrane. So in our analogy of our cell city, this is the city wall, and in it has all these different toll booths, right? And so it has ways to communicate with what's on the outside of the city through our glycoproteins and our glycolipids. We've got stuff that can move in and out of the city through our channel proteins and those other mechanisms that we talked about last week. So this is, again, one of those four things that we find in both prokaryotes and eukaryotes. Controls how stuff moves in and out of the cell. Okay, you guys should be pros at plasma membranes. Okay. So here we go, surface area to volume ratio again. So the way that plasma membrane folds can increase the surface area without changing the volume. And so we can see that again here, this is a cartoon version of the small intestine where we see all those finger-like projections. And we'll see a lot of this when we get into the other organelles. We'll see a lot of these organelles that have these really intricate folding. And it's all about that surface area to volume ratio. Okay. So the cytoplasm then, I like to call the cell juice. So, but in our cell city analogy, this is the actual functioning city itself. This is the space that the city takes up. Okay, prokaryotes and eukaryotes got them. This is where protein synthesis happens. This is, so cytosol. Okay, so here's a potential point of confusion. Cytosol is just a jelly-like goo. Cytoplasm is the cytosol plus all the other crap that's in there. All the organelles, all the solutes, all the proteins, all the stuff. So cytosol is just the juice. And then the cytoplasm is the juice and all the flavorings. Okay, it's mostly water, but it's gooey because it has this high amount of proteins. Also has monosaccharides, disaccharides, amino acids, all the stuff that you need to build a cell. All that, those spare parts are floating around in the cell, in the cytosol, in the cytoplasm. Okay, we got our nucleus. This is the town hall. This is where all the instructions are kept. This is where the DNA is. We want to keep this nice and protected. So we're going to throw a double membrane around our nucleus. And it's so fancy that we're going to even give it its own special name of a nuclear envelope. Okay, so it supervises, houses the instructions for protein production and ribosome production. Okay, this is the instruction center. This is the main frame. Yeah, so. Okay, so double membrane. So the nuclear envelope has that double membrane. It's made of phospholipid bilayers. So whenever you're like, oh, wait, so this is something that I picked up on. I think it's true. But anytime that there's a double membrane, there's DNA inside that thing. So here's our nuclear envelope, double membrane, DNA inside. It has these little holes in the nuclear envelope. Why would it want little pores in there? Get stuff in, get stuff out, right? We make the mRNA is produced in the nucleus. This is the cheap copy of the instructions that we can send out into the cytoplasm. So we got to be able to get it out. We get it out through the nuclear pores. And then it has some goo in their side of there that's called nucleoplasm. And then it's made of this thing called chromatin and a nucleolus. All right, let's talk about chromatin and a nucleolus. First, chromatins. So remember, we talked about how eukaryotic cells have these long chromosomes. Their DNA is this rod shape instead of a circle shape like we see in prokaryotes. So what's happening is that there's these little proteins called histones, and they go and they wind up this long DNA strand so it doesn't get tangled up. Does anybody remember those old-timey, from like way back when I was a kid, those phone cords that had that coil and they get tangled up, and it's such a bitch that we don't want our DNA getting tangled up like that. So the histones allow that wrapping of those DNA strands so they don't get that tangly phone cord business. Yep, so there we go. And this chromatin eventually is what composes the chromosomes. Okay, and then the nucleolus. This is a part of the nucleus where it kind of stains, like when scientists are looking at these things and they stain the cells to see the subcellular structures. This part of the nucleus tends to stain darker because it has so many of those ribosomal RNAs. and it has a lot of those proteins that become the ribosomes. And so that's the same stuff that has to lead through that pore. So that's why we see this dark spot. It's like really producing a lot of ribosomes, the proteins that will become the ribosomes. So it can make proteins. Okay, and we already talked about ribosomes a little bit. These are, in our analogy, these are the production facilities. So we could kind of also think of these as like the individual components of a factory. So this is one of the things that prokaryotes and eukaryotes have. And we might see them stuck to this thing called the endoplasmic reticulum that we'll talk about, kind of like the carrier service of the cell. Or they might just kind of be all stuck together, or they might just kind of be floating on their own. What is the next slide? I can't remember if I. Okay. Yeah. So, but no matter where they are, they kind of all do the same job. And ribosomes have this. There's two subunits. One of them is called the small subunit. One of them is called the large subunit. I really like it when they're given names that make sense, right? So here's the little one and here's the big one. And we talked a little bit about this before, but our mRNA that's produced in the nucleus leaves, goes to the ribosome, docks on, and then we get this the tRNA. That's the guy at the gas station. It's like in the car wash, lines up everything, and then the tRNA, no. Anyway, the other guys, they go and fetch the amino acids, and then that's what builds the protein. So they're found like all over the place, because every cell needs to make proteins, but certain cells tend to have more of them if they make more proteins. All right, here's my little rant about why ribosomes are so important and how. So I study fungi, and I can use ribosomes, the portion of the DNA that forms the ribosomes. This is important. The ribosomes are really important, right? We don't want there to be any mutations, any mistakes in the instructions that build these things, right? And because if you have a mutation that says that messes up the structure of the proteins that produce the ribosomes, you're not going to be able to make proteins, right? So there's a strong evolutionary pressure to get the instructions right. So we see this as part of the DNA code, the codes for these things, that is the same across a lot of different. So that your ribosomal RNA, DNA, in your body is the same as a fungus. So we see this really nice conserved part where there's no mistakes. Because if you make a mistake, you're dead. And then, so there's this chunk, and then there's this chunk that's next to it that doesn't produce proteins. So if there's mistakes here, nobody cares, right? Because it's not going to make anything. So there's this. So what we can do as fungal researchers is say, okay, here I'm going to make a primer, this really short strand of DNA that will dock onto this part of the DNA that is conserved. It's the same across all sorts of different organisms. And then it goes along and it just replicates the DNA. And it's getting into this area where the mistakes can occur, and we don't care. And so then I can sequence that, and I can say, okay, I don't know what fungi are living in my trees, because I'm always getting the DNA from them. And so I can say, if there's DNA from this species that I am looking at in the computer database, then I can say that fungus was in that cottonwood tree. And so I can use the conserved region for this ribosomal DNA to understand the ecological systems and look for fungi that I can't see with my naked eye. I can only detect using their DNA. So that's pretty cool. Any questions about that? All right, so that was my little sub rant about why ribosomes are dope. Okay, so let's see if we can remember the central dogma of biology. It's kind of already written there. So let's review this though, because this is one of those like majorly important things. So we got a bunch of DNA, right? DNA, that's our super valuable stuff. These are our instructions. This is our original copy. Remember the analogy, kind of the story I told about this? Does anybody remember that? Cool. I'm gonna tell it again. So when I was working for USGS, it was the 150th anniversary of John Wesley Powell going through Grand Canyon. And they're like, hey, we should do a thing. And I was like, cool, that sounds good, because I'm a science communication person. I have to do what you say. So I was doing all this research and I found out that at NAU, they housed the original journals that John Wesley Powell and his men kept as they were going down the Grand Canyon. And I was like, cool, I'm gonna read that because that's like the original source, right? And so I went to the Klein Library at NAU and I was like, all right, I want John Wesley Powell, let's go get it for me. And they're like, ha ha ha, no. That stuff is really important. It doesn't leave these highly secure facilities. If you want to look at them, you have to jump through all these hoops. And I was like, well, that sounds complicated. I don't want to do that. So what I did instead is that I went on the internet and I found a digitized copy of that. That if I mess that up, nobody cares because you just print it out again, or you just look at it on the computer and right. So it doesn't matter if this cheaper version of the instructions gets messed up. And so that process of going from DNA to RNA is transcription. We're staying in nucleic acid language. We're just making a cheaper copy that is more disposable. But then the RNA leaves the nucleus, right? If it gets messed up, we don't care, we'll just make another one. Then it has to go through this process through the ribosomes. This is the translation process. This is where we're going from nucleic acid language with our mRNA into protein language with our amino acid sequence. This is the site of translation. So can we put these terms in here, in these boxes? What do you think this is? mRNA, yay, cross that off the list. Okay. This guy. Sorry? So this would be our ribosomal RNA. This is our RNA.
Wait, but I'm sorry. Can you go back one slide?
I think that there was a mistake. Yeah, so I think this has tRNA, but I think that this is supposed to be rRNA because the tRNA's job is to go fetch the amino acids. And so it's not embedded in here. So I think the test is wrong. The tRNA's job is to go grab the stuff. So this is really our RNA. Okay, where are our amino acids at? Yeah, that's little blueberries. It's these little blue circle guys. And then of course, then this whole thing is our growing chain that's going to become our protein, right? So if we can think about the 4 levels of protein structure, where are we at here? Just the order of these amino acids. Primary, secondary, tertiary, quaternary. So I heard primary question mark, And I heard a secondary question mark. If we're looking at just the order of the amino acids, what level is that? That's primary. Yep. And so once it starts to like fold up and kind of interact with the R groups on these amino acids, then we start to get to the secondary. Okay. All right, mitochondria. These are the power plants of our cells. This is where ATP is synthesized. Remember, this is the currency of our city. This is our short-term energy molecule. If we pop off one of those phosphate groups, then we get ADP. This means something diphosphate, right? And then we're going to spend a whole bunch of time talking about cellular respiration, but cellular respiration happens in the mitochondria. So we're taking glucose, we're breaking it down to produce ATP. That's the Cliff Notes version of cellular respiration. Okay, so we have to have oxygen input into the system, and then we can generate carbon dioxide as waste. And then there's Homer showing us how to do cellular respiration. All right. So these rows, guess what? They've got their own ribosomes. They got their own DNA. They got a double layer of their membrane. Hey, they're about the same size as like a bacterium. They got all the same stuff as a bacterium. It's a prokaryotic cell. Oh, interesting. Wonder why. We'll just put that in our back pocket for now. Okay, so our little inner membrane, it folds up a whole bunch to make more surface area for the volume. And then it has what we call these little finger projections, the cristae. And then it has the goo inside, which we call the mitochondrial nucleus. So basically, it's basically a prokaryotic cell jammed into a eukaryotic cell. It has all those same structures and functions. And now it's just producing, the only thing it does is cellular respiration. You guys want to hear about endosymbiotic theory now? Cool. Okay, so the endosymbiotic theory goes like this. Once upon a time, there wasn't eukaryotic cells. It was only prokaryotic cells. And then one bigger prokaryotic cell yoinked up a smaller prokaryotic cell. And instead of breaking it down and turning it into food, it was like, hey, do you just want to like live in here and like, you know, do cellular respiration for me? I was like, okay. So, yeah, sure, take me prisoner, I'll be your slave. It's fine, just don't kill me. So that's how we think that mitochondria came to be, is because it got phagocytosed, but didn't get transferred to the lysosome, and then the other cell was like, hey, this actually works out really good because now I'm freed up to do other stuff. And this guy can just produce ATP for me. And we see the same thing with chloroplasts. Chloroplasts are the same size as bacterium. They have that double membrane. They have their own DNA. They have their own cytoplasm. They have their own ribosomes. They kind of do all the things that a cell does, but it's kind of trapped inside this other cell. Cool thing about mitochondria is you get them from your mom. And so we can actually track the maternal line way back in time. I think I mentioned before that my partner was doing some stuff in the lab, some DNA sequencing, and some skin or whatever flaked off and got into the reaction. And he accidentally sequenced his own mitochondrial DNA, and he found out that he has some Neanderthal blood in him. So, so that's kind of like how we can talk about when we say, okay, well, this is why we know the heritage way back in time is because these mitochondria are passed down through the maternal line. Not in all plants, but in pines, the chloroplasts come from the dads. So we can know the moms and the dads of our little seedlings. And so I can, maybe I'll do like a little blah blah about that sometime, about why it's cool to know who the moms and dads of pine trees are. Okay, so peroxisomes, these are the police of our cell city. These are small round organelles. They only have a single membrane. So if they have a single membrane, there's no DNA for you, my friend. They break down stuff like fatty acids and amino acids. They detoxify poisons. So we like to drink, or I don't know about you guys, but when you drink alcohol, ethanol, all kinds of alcohol are poisonous. It's just that ethanol is the least poisonous of those. So that's the one we drink. And so then that is detoxified in our peroxisomes. They tend to use hydrogen peroxide, H2O2, in order to detoxify those things. So, and then there's all sorts of other kind of oxysomes, depending on what type of cell you are talking about. Okay, and then vesicles and vacuoles. This is how stuff is transported around. So these are the vehicles, whether it's like a little tiny car or a bike or a big delivery truck or a freight train. That's what these things are. And it moves stuff around. So these are membrane bound sacs that have stuff in them. We've already talked about these and how they're formed. So the difference between vacuoles and vesicles, vacuoles are bigger. In fact, in plant cells, these are the major big structure. Takes up 90% of plant cells sometimes, these vacuoles. Vesicles confused with the plasma membrane, or we talked about lysosomes. We talked about how these things are transported in and out and through cells. So vacuoles don't usually fuse with other stuff, but they might house enzymes to break them down. Plant cells don't have lysosomes. Only animal cells have lysosomes. So the job of a lysosome is to break stuff down, but in plants that happens in the vacuole. Okay, and then we'll talk more about the differences between plant and animals later. Okay, so, and then there's all these microtubule organizing centers, and it depends on what these things are, depending on if you're talking about plant cell or animal cell. So I'm just kind of leave it for now. But it does become important when we start talking about cell replication, which can either be mitosis or meiosis, and we'll talk about those later, so don't worry about it right now. And I think that's where I want to end today, even though we still have a bunch of time left. I think I'm going to do the animal cell, the plant cell in the online lecture, so that next time when we come in, we'll just be able to play with these concepts and the other concepts that we'll talk about in the video lectures. Does anybody have any questions? Yeah.
Lab 4 and 5 are due on Thursday.
Lab, so I don't know if the dates are correct on Canvas. So labs are due a week after we finish them. So lab for the plasma membrane lab would be due on Thursday. And then this thing that we did today, that's just to help you study. So there's a way to turn it in if you want to turn it in. If you don't turn it in, that's fine. Yeah, and then just read over the function and properties of cells, starting on page 65. Look over activities 1, 2, and 3. And then I'll either have handouts for you or we'll figure something out as far as what we're going to do on Thursday. Okay. I'll email you guys when I have those videos up on Canvas. And if there's nothing else, then I will see you Thursday.